because I do have a few announcements for to kick us off. So first of all, welcome to everybody. Here we are again for U3A, this time our winter program. You all did such a great job in summer with embracing the technology that I was thrilled to see that the numbers not only stayed consistent, but actually grew for our winter program. So we have had over 145 of you register, which is incredible. We haven't been that lucky in a long time. Believe me, like we're talking decades when we used to do the program on campus. So that is definitely because so many of you are being gracious enough to share U3A with your friends and family, not only locally, but across the country. I know many of you have found us through the article we had in the UT, which was great. So thank you to all of our new friends as well. I'm going to test you now with your Zoom capabilities. So if you are brand new, if this is the first time you're joining us for U3A, can I get a thumbs up or a smiley face or something on by using your reactions on Zoom? So there you go, Gary and Sue, perfect. Who else can we welcome? First timers. If this is your absolute first time, Helen is a first timer, Rebecca is a first timer. So welcome to everybody. Sue, it looks like, Denise, great. Perfect. Dawn, welcome. That's awesome. Rick trying to get in there. He's long-term. Colleen, nice to meet you. So great. Well, that's who I see on my screen, but there's 3 full screens of you all. At the moment, there are 105 of you joining us, which is fantastic. So I should start by introducing myself. I'm Nicole Matthews. I'm the program director for U3A. It's my pleasure to help you navigate the U3A experience. I must say that I had hardly any questions about registering or Zoom this time. So you guys are all learning, you're all doing great. A couple of announcements. All of the sessions are recorded. So we do record the technology session and the speaker. I will break those apart. They come as one big, huge file to me after the day is over, and I will break those apart. So there will be a technology video, and then there also will be the video of the presenter, and I will send that out to all of you much later this afternoon, so you have access to the recording. Know that that's available to you also if for some reason you can't join us live this week, but you definitely want to make sure you still see the speaker. So that's why we do send out the recordings as well. Okay, so that will come to you later today, as well as a reminder about tomorrow's program and a reminder about the Zoom link that you'll need. It will be the same Zoom link all this week. So how you got on today will be how you get on for the next 3 days as well, but I will send that again as a reminder in your email to you later this afternoon. For tomorrow, we do have a really dynamic panel. Hopefully they're dynamic. As you saw in my email that I sent out to you late last week, the topic for tomorrow is about the documentary film called Fandango at the Wall. Has anybody been able to watch the video yet? Yeah, a few hands. Great. Okay, good. So, so those of you who have seen the film, some of the main participants, I don't want to say characters because it was a documentary, but the main people that they feature are all going to be a part of a panel discussion with us tomorrow. So I would highly recommend, if your day permits it, it's about a 90-minute film for you to watch the film if you can today, just so that I feel like the discussion tomorrow will be a lot richer for you if you've actually seen the film. I will again send out the link for you to do that. We were able to find it by just doing a search on by Amazon Prime through our Prime app on the TV. We did a search for it and we found it there. So we didn't have to use that link that I sent you. Doug mentioned to me previously that it was also available on HBO. So it should be free to you. There's no cost for you to watch it, but I just want to let you know that that is available to you. Just as a reminder, we had a great turnout for our tech workshop this morning. We had about 85 of you that joined us for tech, so that's fantastic. If you are more than welcome to join us tomorrow for tech, it runs from 8:15 to 9:15 every day. that we have the program. Rick generally jumps on around 8 in the morning, so a few minutes early. So if you have a really specific question that you want to kind of get a one-on-one tutoring with, he's available at 8 for you to do that. But then at 8:15, we'll do a kind of a larger session. Okay. And then the last thing is just a reminder, I did have a couple of you, actually more than a couple, that when you entered your registration, you didn't put in the right email address. So you maybe aren't getting the information that I've been sending out. That's because I'm sending to the email address that you have entered in. So just as a reminder, I know sometimes our fingers get a little bit big and we hit the wrong key, but if you don't give me the right email address, I can't send you the information. So if you are relying on your friends right now to send you the information because you have not been receiving it, please make sure that you reach out to me, email me today so that I can update your email information so that we don't, so that you don't miss any key information. Okay. And then my last point is just for those of you who are longtime U3Ers, does anybody know Marta Brown and is she still well? I've tried to reach out to her. I'm not hearing from her, so I just want to make sure she's okay. So if anybody is personal friends with Marta, if you could just let me know how she is and hopefully she's still well, that would be great. Also, just a final reminder is if you do have your camera on, just remember we can see everything you are doing. So this is not a time to change your clothes. This is not a time to get out of the shower. You think I'm joking, we've seen that. So just as a reminder, whatever is happening around you and behind you, we can see. So just be mindful of that if you're on camera. If you feel like you need to change your clothes during the presentation, just turn off your camera just so that the rest of us aren't enjoying that show. We'll enjoy that at a later time. Okay. So we'll leave it at that. At the end of the presentation, we can chat about some more housekeeping notes for the rest of the week, but I don't want to delay Dr. Bird in his presentation. So you all received the bios for all of our speakers. Dr. Bird is one of our professors here at USD. And I think there's no better time to talk about pandemics than the one we're living through, but he's going to take us back to pandemics in the medieval times. So Dr. Bird, thank you so much for being with us today.
Thanks, Nicole. I'm going to go ahead and share my screen. Hopefully you can all see PowerPoint.
You're looking great. Good. Thank you.
All right. And let me minimize something here. Okay. As Nicole said, these times that we're going through right now, I guess I don't really have to define what a pandemic is. All of us have seen how quickly disease can spread from one part of the globe to another. Imagine what it was like several hundred years ago in medieval times, where transportation wasn't as great, news didn't travel as fast, and pandemics could appear out of nowhere. We see that art and sculpture in medieval times and later were dramatically affected by these kinds of experiences. This is one of my favorite paintings, The Triumph of Death by Bruegel. I believe this was painted back in the 17th century. After Europe had experienced several pandemics that would appear from time to time, here the pandemic is depicted as an army of skeletons laying waste to a city, and no one is safe from these pandemics. As you can see, a monarch down here in the corner is being dispatched by a skeleton while another helps himself to the king's gold. These were frightening times, and they had dramatic effects on Europe and other parts of the world. What I want to do is concentrate on a particular pandemic that probably all of you are aware of. Let me see if I can advance my slide. The bubonic plague. The bubonic plague that you've probably heard about is sometimes referred to as the Black Death, which struck Europe in the 14th century. And it was a devastating pandemic that killed, some estimates go as high as a third to a half the population of Europe. It was, many of you may know, it was characterized by the appearance of large buboes that were major swellings that would occur mostly in the armpits and the groin area, that would become discolored. And the poison that was in those buboes would spread throughout the body, and death was very, very fast, frighteningly fast. For people, in a matter of 2 or 3 days, the victim was usually dead. There were actually 3 pandemics of the bubonic plague that we know about. There may have been more. Research is looking back into further times. But the 3 pandemics that we know were caused by bubonic plague began about 1500 years ago with the Justinian plague, which struck the Byzantine Empire. It struck Constantinople in 541. And that plague, there's some people who believe that that plague then went through successive waves throughout the Mediterranean and may have lasted for more than 200 years. The Black Death, which most of you are probably familiar with, struck Europe in 1347. It spread from Southern Europe up through Northern Europe and into England. And as I said before, it may have killed up to half the population of Europe over a period of time. And then finally, the modern plague, which began a little over 100 years ago, or 150 years ago, in China and spread throughout Asia and across to North America. I'll touch on all 3 of these plagues, spending most of my time talking about the Black Plague. And then what I'd like to do, since I'm a microbiologist who's interested in bacteria, we'll talk a little bit about, or I'll talk a little bit about the bacterium that is responsible for the Black Plague, where it came from, and how it acquired the ability to cause pandemics. As I said, the plague of Justinian struck in 541. It's believed that the disease arrived from Asia and made its way into Europe through existing trade routes at the time. And in 541, trade between Egypt and Constantinople, which was the capital of the Byzantine Empire, that was the eastern half of the former Roman Empire. The disease arrived in Constantinople, and within a very short time, within a period of a couple of years, spread throughout the Mediterranean. Trade routes, shipping routes are believed to have allowed the disease to spread through areas, countries all surrounding the Mediterranean, including Northern Europe. Within a fairly short time, 25 million people are estimated to have died. And as I said before, researchers have found evidence for multiple waves, perhaps as many as 18 waves of bubonic plague rolling back and forth over this area, decimating the population and killing as many as 50 million people by the time it disappeared, possibly as late as the 700s. The Black Death, which we usually think about when we think about the bubonic plague, is believed to have begun with a Mughal army that arrived in what is now the Ukraine at about that time. There was a city called Kaffa, which was administered by Genoese merchants. And a series of events led to the Mughal army laying siege to that city, trying to capture it. And at that time, bubonic plague broke out through the Mongol army. And within a very short time, the Mongol army was reduced to the point where they realized they were never going to be able to capture the city. But before they left, there are reports, I don't know how accurate they are, they're from a historian that we know lived at the time but was not in Kaffa at the time, who recorded the fact that Mongols are believed to have loaded up corpses of soldiers who had died of the plague onto catapults, catapulted their dead bodies over the walls and into the city, If so, this may be the first instance of biological warfare that was ever used. In any case, as soon as the siege was lifted, the Genoese got on their boats and sailed back into southern Europe. They first attempted to land at Sicily, but by that time bubonic plague had broken out on the ships, bringing the Genoese back. They were not allowed to disembark, but they were in port long enough for the disease to jump into Sicily. The sailors sailed on to Genoa, where they were able to disembark, and of course the disease joined them or disembarked with them in their capital, Genoa. And from that point began to spread throughout Southern Europe, and over the next couple of years, slowly progressed northward through France, and eventually jumped the Channel and entered into England. Over the next 7 years, the bubonic plague moved from city to city. Some of the cities that were especially hit hard were Florence and Vienna, where as many as a half or more of the city's population were killed. The disease typically would eliminate all of those that were susceptible to the disease. Very few people, as perhaps 20 to 30% of the people were able to survive bubonic plague. And if they did, if they were lucky enough to survive, they became immune to the disease. The disease would move on to other major populations. And after 20 or 30 years, the disease could then come back to a city that had already devastated, because more people had been born. Because you can't inherit immunity to bubonic plague, those younger people would be susceptible, and the plague would take new victims. There's still controversy over how the plague was able to last for a few hundred years in Europe. It's possible that the plague simply was able to rotate around through major cities whenever new susceptible people were born. But it's also possible that multiple successive waves of bubonic plague arrived out of Asia at various points to strike Europe over the next few hundred years. As for the cause of bubonic plague, nobody during the first two pandemics had any idea of what was causing the plague. The most prevalent hypothesis was the idea of miasma, the idea that bad odors cause disease. So exposure to rotting corpses was thought to be a source of disease. And there was recognition that once the disease got hold in a population, that it could be contagious and that the disease could move from person to person. And so quarantining became a common and was a somewhat effective way of keeping the disease from spreading outside of cities. But no one really knew for sure what was causing the disease. A couple of hundred years later, after the initial outbreak of the Black Plague, the last time London was struck by the plague is referred to as the Great Plague, where somewhere estimates put the number of people who died between 70 and 100,000 people, roughly 20% of the population. As I said, most people felt that miasma was responsible for these sorts of widespread pandemics. And so throughout London during this time, major fires were lit as a way of trying to purify the air and reduce the effects of the pandemics. Many of you may have seen images like this at the top right of doctors that would try to treat people during the Great Pandemic, covered with a heavy cloak and gloves that were meant to try and protect them, a wand of incense, and sometimes pockets full of flowers were worn by these doctors as a way of having the nice fragrances from combating the miasma that they felt was causing the pandemic at the time. We actually have a fair bit of information data that tells us that the number of people who died during the Great Plague is accurately known. At the time, bills of mortality were used they were published weekly as a way of keeping tabs of how many people had died the previous week and what had caused their death. These were kept primarily to warn the population when a new plague of some kind, whether it was smallpox or bubonic plague or tuberculosis, whenever a new pandemic was rising, these bills of mortality would hint at that. This particular week comes from the height of the Great Plague that struck London in 1665. And you can see there was a number of deaths here attributed to things like consumption or tuberculosis, spotted fever, but the vast majority of deaths here, out of a number of burials of 8,200 or almost 8,300 deaths, the vast majority of them, 7,165, were believed to have been caused by the plague. And so we have a pretty good idea of the number of people that were dying of plague in London. I might just draw your attention to how detailed some of these bills of mortality are. There was one poor individual killed by a fall off a bell tower. And so although we're not sure exactly how accurate they were in terms of diagnosing the disease, at least the numbers of deaths that were occurring at the time are probably pretty accurate. This chart reminds me of some of the things that we've all been seeing on the news lately with the waves of the current COVID pandemic that are striking different parts of the world. The plague of 1665 that struck London struck as a very, very rapid wave that occurred towards the end of the summer in 1665. The solid line here marks the number of deaths that occurred each week throughout that time. The dashed line below are the number of deaths that occurred to bubonic plague. And so you can see that by August, end of July, August, there is a tremendous increase in the number of people dying of the plague. It lasts for a couple of months, and then very, very rapidly, the number of plague deaths drop off, so that by the time the winter approaches, the rate of mortality is just about normal in the city. There's a couple of things to take away from this chart of the pandemic. It has two major hallmarks of the pandemic. One is the incredible mortality that occurs. In this case, bubonic plague, we'll see in a moment, there were multiple forms of bubonic plague, some with mortality rates as high as 90 to 100%. But the other thing that is striking about this is just how quickly the pandemic wave appeared and disappeared. What this indicates is that this particular pathogen was incredibly contagious. It was very, very easy for the pathogen to jump from victim to victim. As we're seeing with COVID, moving very quickly through populations, this pathogen that was causing the bubonic plague is able to move very rapidly from one victim to the next, resulting in this very, very sharp increase in deaths and this spectacular wave that occurred in the summer of 1665. We'll come back and talk briefly about the medieval Black Plague a little bit later, but I want to now just turn our attention to the third pandemic, which struck China in the middle of the 19th century. And this particular outbreak is responsible for killing over 12 million people over the next several decades, primarily in Asia, India, and Pakistan, but also moving across to North America and introduced bubonic plague to North America. This particular plague, because it happened in the latter part of the 19th century, coincides with the emergence of germ theory. As I said, most people associated contagious disease with miasma, the idea that bad odors could cause disease. By the end of the 19th century, microbiologists like Louis Pasteur and Robert Koch developed germ theory, the idea that microbes, which had been known for a couple of hundred years before, that microbes were actually the root cause of contagious disease, and that specific diseases like smallpox, like bubonic plague, were caused by specific microbes that could be treated. And so in the latter part of the 19th century, many of these scientists worked to study particular diseases and try to identify the particular microbe, whether it was a virus or a bacterium, that was causing the disease. Hong Kong was struck by the bubonic plague and the third pandemic in 1894. And Louis Pasteur sent one of his best scientists, Alexander Yersin, to Hong Kong to see if he could identify the microbe that was thought to be causing bubonic plague. Alexander Yersin was Swiss, but he was a French Swiss. And when he got to Hong Kong, which was governed by the English, initially hospital officials were not interested in collaborating with a Frenchman. And so Yersin had to bribe officials at a hospital to get cadavers of people who had recently died of bubonic plague. But once he got them, he was able to dissect the buboes in these cadavers and was able to identify a bacterium that's shown down here in the center, the lower part of the slide here, a rod-shaped bacterium that now bears his name, Yersinia pestis. I guess that's what happens when you discover a deadly bacterium, you have the honor of having it named after you. This bacteria, as I said, was isolated from the tissues of buboes of people who died, and he was able to show that this was the microbe that was actually causing the disease. He also hypothesized that rats were somehow carrying the pathogen, and that the bacteria was able to spread from the rats into the human population. And I think one of the pieces of evidence that led him to that hypothesis was that many times when a major population, a human population, was about to be struck by bubonic plague, there would be a major dying off of rats. As the bacterium was killing off the rats, it was then somehow jumping from the rat population, which had been destroyed, into the human population, where it would cause bubonic plague pandemics. Yersin's hypothesis was confirmed a few years later by Paul-Louis Simond, who was able to show that fleas were able to carry the bacterium. And as you're probably aware, this is the basis of bubonic plague, the Black Death in Europe. It's thought that rats were able to spread, that carried the bacterium, spread throughout Europe, and that fleas then were transferring the bacteria from rats to humans, causing the pandemic. So in addition to identifying the microbe that was causing bubonic plague, further studying of the disease led scientists to realize that there were three major forms of the bubonic plague. The first of these, the most recognizable, the bubonic plague itself, is characterized by the emergence of these large buboes, swellings of lymph nodes that have become infected with the bacteria. It's been shown that fleas that carry the bacteria from infected rats, when they bite a person, inject the bacterium Yersinia pestis into that person, the bacteria then are able to make their way to lymph nodes, which are a structure that the body uses as part of the immune system to fight off disease. But instead of being able to stop this disease, the lymph nodes actually house a large population of bacteria, which are able to grow inside the lymph nodes, causing hemorrhaging, which results in the blackening of the lymph nodes. These nodes enlarge, they're pus-filled, full of growing bacteria, increasing numbers of bacteria that eventually break out of the lymph nodes, move throughout the body in the bloodstream, destroying tissues, resulting in death about 50% of the time. But in addition to the bubonic plague, there is a pneumonic form of the plague, where individuals who maybe are in the later stage of bubonic plague have infected lungs. When the lungs are infected by the bacteria and tissue is destroyed, the victims begin to cough up large droplets of blood that contain bacteria. If those bacteria-laden droplets are inhaled by somebody else, they can contract bubonic plague directly through their lungs. In this case, it doesn't require a flea bite to transfer the bacteria. This is a very serious form of the plague, which can kill people within about 48 hours and has a much higher mortality rate of up around 90%. And finally, a septicemic plague can occur. Generally, this occurs in the latter stages of bubonic plague, but it can also occur if a flea bite bites directly into a major blood vessel, transferring a large number of the bacteria directly into the blood vessel. These bacteria rapidly spread throughout the body and attack tissues and lead again to a very rapid decline in death. And the mortality rate is virtually 100% at this time. And so what became clear to researchers is that Yersinia pestis, in addition to being able to be a very, very deadly pathogen once it gets into the human body, has some special properties that allow it to be able to use the flea as a vector that move the bacteria from infected rodents like rats, ground squirrels, into the human population. Now we know that the third pandemic was caused by Yersinia pestis because, as I said, Alexander Yersin was able to isolate the bacterium from people who had recently died of the plague. But what about the first and second pandemics that happened 1500 to 800 years ago? Before we realized that bacteria could cause this disease, how do we know for sure that those two pandemics were caused by Yersinia pestis? Well, fortunately, DNA molecules are very, very stable molecules. All living organisms, including bacteria, use DNA as their genetic material. And in the last, 10 to 20 years, researchers have gotten very, very good at isolating DNA molecules from ancient bones and skeletons. And these DNA molecules can be analyzed, they can be sequenced to determine the kinds of microbes that were present in the person's body when they die. And so what researchers have been able to do, particularly in the last 10 years or so, is to analyze skeletons from plague pits that date back to the Justinian times, about 1500 years ago, or to the Black Death 600 to 800 years ago. And researchers can analyze and extract microbial DNA from these skeletons, sequence the DNA to identify the microbes that were present in those bodies. Typically, what researchers will do is use teeth, where they can drill into the teeth. The teeth have a pulp inside and blood vessels inside that is protected from decay once the skeleton has been subjected to microbes in the soil after burial. The DNA within the tooth is largely protected from these sorts of degrading, decaying microbes. And any bacteria, any pathogens that were present in the body that were circulating through the blood would have its DNA preserved inside these teeth as well. And so what researchers have been able to show now is that bodies from Justinian plague pits or from the Black Death medieval time plague pits, both of these skeletons, both of these plague pits do indeed carry DNA from Yersinia pestis. And so we know that bubonic plague is responsible for the first and second pandemics, the Justinian and Black Death pandemics. And so what I want to do now is turn to the biology of the pathogens. What is it about Yersinia pestis that allowed it to be such a devastating pathogen. Of course, we're watching the news and reading the news over the last several months, trying to understand how a virus could cause COVID. What is it about the SARS-CoV-2 virus that allows it to cause such a deadly pandemic and widespread pandemic? Researchers have been doing this for Yersinia pestis to try to understand how it was able to become such a deadly pandemic or pathogen. Now, interestingly, this research has showed us that Yersinia pestis is a relatively new bacterium, a new species of a bacterium that has appeared in the last 10,000 years. Its most closely related cousin is a bacterium called Yersinia pseudotuberculosis. Yersinia pseudotuberculosis is a soil bacterium primarily, but it's capable of infecting ground rodents, burrowing rodents, like paradox or ground squirrels, marmots, which are a type of ground squirrel. Any of these rodents that burrow in the soil have the potential to pick up these bacteria. And these bacteria are generally bacteria that can infect the gastrointestinal tract of these rodents, and occasionally infect the blood and circulate through the blood of these rodents. Yersinia pseudotuberculosis is still present today, and it can cause mild gastrointestinal infections in people, but generally they're very mild, and very often people don't even realize they're infected by these particular bacteria. But this particular bacterium is limited to infections of the gastrointestinal tract. But we realize now, what we've learned now is that Yersinia pestis diverged from this bacteria less than 100 years ago, and less than 10,000 years ago, and acquired the ability to cause blood infections in people, leading to deadly pandemics. And what we've been able to do now is chart a series of genetic changes that have occurred that allowed Yersinia pseudotuberculosis to gradually change into another bacterium, Yersinia pestis, that was capable of pandemics. Recently, scientists have been able to extract Yersinia pestis DNA from skeletons of people who lived during the Bronze Age. So there are a number of sites here. There are 6 or 7 sites throughout Eurasia that date back as far as 5,000 years ago. And researchers have been able to extract Yersinia pestis DNA from these individuals, from some of these individuals, indicating that they died of bubonic plague as far back as 5,000 years ago. Now, what's interesting about this is a number of things that are interesting about it. One is that we realize that this form of Yersinia pestis that lived 5,000 years ago could not use fleas to move from person to person or from rodents to people. So most likely these people died of bubonic plague after they killed and ate ground rodents like marmots. Marmots are still killed and consumed in some parts of Asia. They're large, meaty rodents that provide food, and they have nice pelts that can be used. But what's suspected is that occasionally a hunter killed and consumed a marmot that was infected with these early forms of Yersinia pestis. They contracted bubonic plague. They may have been able to transfer it to other, or transmit it to other people through the pneumonic form, through the lung form of bubonic plague. But because the bacteria was not able to use fleas, it was not able to cause a major pandemic. And so these were largely, is thought to be largely smaller outbreaks that killed a number of people after people hunted and consumed infected rodents. So this, all of these Bronze Age infections occurred before the bacteria was able to use the flea as a vector to be able to transmit from one human host to another. So what is it about fleas that allow for the rapid spread of a bacteria through a human population. Well, if you think about it, this is very similar to mosquitoes being able to spread malaria. Fleas, like mosquitoes, take a blood meal. They then digest blood, whether it came from a rodent or from a human, as a food source. If the flea bites a rodent that's infected with the bacteria, so in addition to sucking up red blood cells, it's going to suck up the bacteria. The bacteria then could be capable of digesting and eating the red blood cells that are part of the flea's meal. That allows the bacteria to grow its population inside the flea. And then when the flea goes to bite a second host, if it regurgitates a little bit of its meal, it's going to regurgitate some of the bacteria into the new host. And this, of course, allows the bacteria to move from one rodent host, perhaps, to a human host. The flea that's pictured here is Xenopsila chiopus. This is a flea that prefers to feed on rats, particularly the black rats. But it is known that when there is no rat host, these fleas will then turn to humans and take blood meals from humans. And so this flea is thought to, the species is thought to have been a major player in allowing this particular bacterium to spill over from the rodent population into a human population. Now, what I want to do is take a little bit of time and talk about how scientists study the properties of bacteria that allow them to become effective pathogens, and focus on how we know, or what we've learned about how Yersinia pestis was able to evolve into a pathogen that could use the flea as a vector to move from one rodent to another, or from an infected rodent into a human. So up top to the right here is a diagram of a flea. We have the head part that would take the meal, an esophagus, leading down into what's circled here is the proventriculus, is a muscular structure that is at the end of the esophagus and empties into the midgut. And it has a very important function as a valve. It's a one-way valve that allows fleas to take a blood meal in first one meal, and then to take a second blood meal. What the valve does, the one-way valve does, is allow for a first blood meal to enter the gut, but will not allow any blood that was taken in a previous meal to be regurgitated while it takes a second meal. And so it's a one-way valve that lets material into the midgut, but not allow it to leave the midgut. And the midgut is where most of the digestion occurs. And then there's a hindgut behind that allows for the excretion of feces once digestion is complete. And so what researchers have been able to do is take Yersinia pseudotuberculosis and study how it is able to infect the flea, or whether it's able to infect the flea. So this is a microscopic image of a particular flea. Here's the esophagus, shown by the E, the proventriculus, this structure that is at the top of the midgut, the large midgut, and then the hindgut is where feces are excreted. When a flea is infected by Yersinia pseudotuberculosis, the ancestor to Y. pestis, it's only able to infect the hindgut. Here, the bacteria, the large population of bacteria, has been stained green. And so we see these bacteria are able to colonize the hindgut, but they're not able to colonize the midgut, where most of the blood of a previous blood meal is sitting, and they're not able to colonize the proventriculus. Although they can colonize the gut, they can't grow into a large bacterial population, and they would only be shed by the flea through feces. And so while it may be possible for the bacteria to move from one rodent to another using the flea as a vector, it would be inefficient only if other rodents came into contact with infected feces. So this is the normal, or what we call wild type Y pseudotuberculosis property. But bacteria have a special property that allows for genetic information to move from one species of bacteria into another. It's called conjugation, or sometimes referred to as bacterial sex. Although it's not really sex, it has nothing to do with reproduction. It is simply a way for genetic material to move from one species of bacteria into another species of bacteria. Some of you may have heard or read about the rapid spread of antibiotic resistance that is occurring in bacteria. One of the reasons for that is that different species of bacteria can share genes that confer antibiotic resistance, or resistance to some of the drugs that we use to try and kill bacteria, bacterial infections. Conjugation is a process that allows genes to jump from one species to another. So we now know that an early ancestor of Y. pestis acquired new genetic information from bacteria that were able to colonize the midgut of a flea. This new information is believed to have been acquired through conjugation and allowed Yersinia pestis to now colonize the midgut. To test that hypothesis, researchers have taken a single gene. It's called the YMT gene, and we won't even talk about what it does, but when this little bit of DNA is inserted into the Y. pseudotuberculosis genome, now this new form of the bacteria is able to colonize the midgut. And we see the glowing green bacteria able to colonize the midgut. Now this is important because it means now the bacteria can feed on the blood digesting within the gut of the flea, and allows the bacteria to grow their population. But the bacteria can still only leave the host through feces, making the flea still somewhat inefficient in terms of being used as a vector. Another series of mutations. Now these are mutations now where the mutations have changed existing genetic information in the bacterium itself. In this case, what it did was resulted in the loss of control over something called biofilm formation. Something we've learned about in the last 20 or 30 years is that many bacteria have the ability to form into dense assemblages of bacteria embedded within a sticky material that allows them to live attached to a solid surface. Most of the time, bacteria will only do this when they're living in a natural environment, not when they're living inside of a living organism. And bacteria are able to control when they live as a biofilm or not. They have a number of genes that act to shut off other genes that are necessary for building a biofilm and turn it on only when it's advantageous. These particular mutations that occurred in Yersinia pseudotuberculosis removed the control of biofilm formation. And what this did was allow Yersinia pestis now to not only colonize the proventriculus ahead of the midgut, but to also live as a biofilm within this structure. Now the proventriculus in an uninfected flea looks something like this. These finger-like projections are able to allow the proventriculus to work as a one-way valve, as I mentioned, where material can enter the midgut, but they can't leave the midgut and get back up into the esophagus. But when the Yersinia pestis grows as a biofilm in the proventriculus, it disables that function. And so bacteria are now able to move in both directions, into the midgut and out of the midgut and back up into the esophagus. Now this had major consequences because it means that when the flea takes a blood meal that might, from an infected rat that would take up bacteria as well as the blood meal, the bacteria feed on the blood meal inside the flea. And then when the flea takes a second blood meal, because this valve in proventriculus is no longer working, it regurgitates its previous meal into the new victim. And that includes a large number of bacteria, up to 25,000 or more bacterial cells can be injected into the new victim. And so now we have a very effective vector that allows the bacteria to grow its population in the flea and then use the flea to move from one infected rodent into an uninfected rodent or from an infected rodent into a person. Not only is it an effective vector, but if you think about it, every time this flea tries to take a blood meal, it loses its previous meal. And because it's lost its meal, it begins to starve. And what is a starving flea going to do but keep biting? In fact, its bite rate increases dramatically as it desperately tries to acquire a new blood meal and digest it. Eventually, these fleas generally starve to death because they can't retain a previous meal due to the inactivation of the proventriculus. And so we see that in this experiment here, where here's use pseudotuberculosis with the same mutations that allow for the biofilm formation to occur. This bacteria now is not only able to grow a large population in the midgut, but is able to populate the proventriculus, allowing it to use the flea as a very effective vector to move from rat to rat or rat to person. And so just to summarize what I've told you, I know there's a lot of information there, and we can come back and chat about this at the end of the lecture if you like. But just to summarize, there was a major gain of genetic information through conjugation that allowed an early ancestor of Y. pestis to move from being only able to colonize the hindgut to being able to colonize the midgut and digest the blood meal that the flea had taken. A loss of genetic information, the ability to regulate that biofilm formation, allows the bacteria to move up into the proventriculus and activate its one-way valve function, turning this flea not only into a good incubator of bacterial growth, but as a very good and effective vector, moving the bacteria from one individual, a rodent or person, to another. One more mutation I want to tell you about, and then I'll stop talking about the genetics, I promise. And that is the gain of another gene. This gene called PLA was also acquired through conjugation, it's believed. And what it does, this gene codes for an enzyme that allows for the bacteria to degrade blood clots. One of the ways the human or the rat body tries to protect itself from abrasions to the skin is for the blood, for platelets circulating through the blood to create a clot that not only stops the loss of blood through the injury or the bite, but forms a dense wall that traps any pathogens at that site and will not let it disseminate throughout the body. What PLA does is it's secreted by the bacteria and it digests this blood clot, breaking down the blood clot and allowing the bacteria to spread from the site of the flea bite. This is a microscopic image of a skin of a mouse that has been bitten by a flea 3 days before. These dark spots here are the bacteria that have been able to move. This is 3 days after the bite. This is an image of the flea head with this proboscis in the flea bite. And what PLA does is allows the bacteria to break out of the initial bite, be able to move through the body, get to the lymph nodes, as we talked about, and cause the growth of those buboes at the start of bubonic plague. And so we know now that it's actually been a series of mutations that occurred in Yersinia pestis over a few thousand years that allowed the flea to serve as a vector for spillover. Spillover is the term that's used to describe when a microbe is able to move from one species of animal to another. In this case, the rat flea is able to be a vector that allows for the bacteria to spread from one species of rodent to another, to essentially cause a pandemic in rodents, mostly ground rodents. And the flea also has the capability of transferring the bacteria from these rodent populations into humans, causing bubonic plague, where the flea is able to bite. The PLA gene digests the blood clot, the bacteria able to move throughout the body to the lymph nodes, causing buboes to occur. The same plagine that allows the bacteria to break out of blood clots, we now know is required for the bacteria to be able to cause pneumonic forms of the plague, airborne forms, where the bacteria is inhaled into the lungs. The plagine allows for the bacteria to begin to break down lung tissue, causing hemorrhaging and allowing the bacteria to gain entry into the bloodstream of its unfortunate victims. So this is the phylogeny of Yersinia pseudotuberculosis. A phylogenetic tree like this is really very much like a family tree, where over here on the left, we've got ancestors. Over here on the right, we have more modern versions of different strains of Yersinia pestis. So time is moving from left to right, far back in time on the left side, more modern times on the right side. And what this tree or branching depicts is the occurrence of different genetic mutations. We know when they occurred by analyzing microbial DNA that came from skeletons from these plague pits. It tells us the sequence of mutations when and where they occurred that allowed for pseudotuberculosis, Y pseudotuberculosis, to produce a new pathogen, Y pestis, Yersinia pestis, that was able to sicken people back in the Bronze Age. Here they were able to sicken people in the Bronze Age, but they had not yet acquired these mutations that occurred less than 3,700 years ago BC. These mutations here allowed for the bacteria to use the flea as a vector. So now that leads to the ability to cause the plague of Justinian back around 541. A second mutation is thought to have been an important player in that ability to cause that pandemic. The bacteria acquired the plagine almost 7,000 years ago, was able to cause pneumonic plague, but a mutation in the plagine after it got it, a small mutation, allowed it to become very, very effective at breaking down blood clots, allowing the bacteria to spread through the human body. And this has been referred to as the Big Bang, as in the evolution of Yersinia pestis, to be able to cause the three major pandemics that have occurred since then. This mutation wasn't present in the bacteria that caused the Bronze Age plagues. And so it's thought that that's the reason why these never were able to cause pandemics. So the origin of Black Death, we believe that Yersinia pestis had evolved from Yersinia pseudotuberculosis. It acquired the ability to infect ground rodents like marmots, seeing here, spread through large populations of marmots. It's thought that different seasonal changes that may have occurred at various times allowed for explosions in rodent populations, allowing more contact between rodents and people, infected marmots that may have been killed by Mongols, who we know consumed marmot meat and used their pelts, allowed the bacteria to gain entry to the human population. Mongols, because they had large, very mobile armies, would have been very effective at moving the bacteria from Asia, different parts of Asia, throughout Asia, and into Europe, where the bubonic plague was able to establish itself. Today, we know that there are endemic populations of rodents that are infected with Yersinia pestis. So on this world map here, anywhere where you see red, we know that there are wild populations of ground squirrels, gerbils, marmots that are currently infected with Yersinia pestis. And they have the ability, periodically, these bacteria have the ability to spill over into humans. And you'll see that you can see the countries that are colored in yellow are countries that have reported bubonic plague within the last few decades. And of course, you can see in the United States, it has a very large foci of rodent populations in Western United States that we know carry Yersinia pestis. We know that these bacteria arrived in North America from China by way of San Francisco. The bacteria was able to spread into the rodent population throughout Western United States, and periodically cause bubonic plague in the United States and other countries. These are some of the headlines that I have pulled from the internet. Some of these are from Mongolia, but also from the United States, including Colorado, where bubonic plague pops up every now and then, where somebody comes into contact with a rodent. Occasionally, marmots are still eaten in Mongolia or parts of China. There was a sad case a couple of years ago of a newly married couple where the groom killed and consumed a marmot and shared it with his new bride. It is traditional to eat part of the marmot uncooked. This allowed the bacteria Yersinia pestis to infect those people, and unfortunately, they die. Over the last few years, we've seen annual pandemics in Madagascar. The bacteria has gotten into rodent populations in Madagascar, and in the summers, bacteria have been breaking out and causing a few hundred deaths each summer. Researchers are working to identify which rodent populations have these bacteria and to try and get antibiotics quickly to these people who are infected to try and save their lives. But ominously, a few years ago, we started to see the first antibiotic resistant strains of Yersinia pestis begin to pop up in Madagascar. So that's a very serious situation. Occasionally, we'll hear of a bubonic plague or Yersinia pestis being found in rodent populations in Yosemite Park. Plague-infected squirrels were found there a few years ago and resulted in the closure of some of the campgrounds. And so, although we think of bubonic plague as being an ancient disease that killed millions of people, but in the distant past, we have to realize that this bacteria is still present in many parts of the world and requires great care to make sure that it doesn't get into the human population. So if you are visiting Yosemite Park or some of the other national campgrounds in the western part of the United States, I recommend that you don't feed the squirrels. Otherwise, there's no guarantee that you won't experience the same pandemic that people suffered from in the bubonic plague. And I'm going to stop there, and hopefully you found this of some interest and that you have some questions for me.
Thank you, Dr. Bird. That was really interesting. And we do have some questions in the chat room, so I will facilitate those for you.
Okay.
But my first question would be, if you were giving this presentation in 100 years, what would you be adding into the presentation about our current situation with COVID and how it stacks up with everything else you've shared?
Yeah, well, that's a great question. And I think I've included another slide here. Let's see if I can move that. I actually anticipated a question like that. And I think a lot of people have been thinking about that. Here's another phylogenetic tree like the one I showed you about Yersinia pestis, but this one is for SARS-CoV-2. This is the virus that causes COVID-19. And what researchers do nowadays is travel throughout the world, isolating samples from various wild populations of animals, especially bats, because bats are known to carry a number of, especially viruses that can infect people. And what they do is analyze these sequences, so these DNA sequences that they get from viruses in these animals, and keep tabs on them. So this particular phylogeny shows the phylogeny of, here's the virus that causes COVID-19. And what people are trying to do is figure out where it came from, how it evolved. So much like I just described, we're trying to learn how Yersinia pestis evolved into a deadly pathogen that's capable of infecting people. We're trying to understand where this virus came from and how this virus has evolved into something that it can jump from bats and people. And just as importantly, how it's able to spread so quickly from person to person. Um, you've probably heard that there's a new variant of the virus that has emerged within the last month or so. This is the second variant that I know about that has come about since the pandemic began almost a year ago. And so what we've learned is that mutations that are affecting the structure of these spike-like images, people are probably seeing lots of pictures now of the virus that causes COVID. It has these spikes, mutations that alter these spike proteins appear to be making the virus even more infectious, not more deadly, but more infectious and able to jump from person to person more efficiently. And so in the next few years, hopefully sooner than 100 years, what we'd like to know is where this virus came from, how it acquired this property that allowed it to jump from bats to people, and how is it acquiring the ability to increase its infectivity to be able to move through populations so quickly.
Yeah, good, interesting. Okay, great. All right, so if you do have a question, please drop it into the chat box. So here we go, rapid fire questions, if you will. All of these pandemics, as well as coronavirus, seem to originate in Asia. Thoughts on why this might be so?
That's a good question. So bubonic plague, the bacteria there is thought to have evolved from an organism, as I described earlier, as from a soil bacteria that is able to infect ground rodents. So ground rodents, when they're digging burrows, able to spread into human populations from there. I don't know that this had to be China, whether there's anything special about Asia or China that allowed for this to occur, except that eating ground rodents in Asia has been done for thousands of years. And so it provided an opportunity for the bacteria to initially jump from rodents to people through frequent consumption of those animals. In terms of the virus, like SARS-CoV-2, Asia is home to many, many bat species. Large numbers of bat species live in that area. And bats are the perfect reservoir for viruses because they live in large populations, very dense populations. They tend to groom each other. And so they're a perfect animal reservoir for a microbe like a virus to be able to evolve. And then bats are consumed in Asia from time to time, allowing for close contact between those kinds of animals and humans. So that's one of the reasons why we think a number of these diseases are able to or tend to be traced to China. Influenza is another one. Influenza evolved from birds, from an avian flu. And anytime large numbers of birds are kept with large numbers of people, there's the opportunity for pathogens to jump from a bird population to humans. And again, in Asia, we have large numbers of farmers farming fowl, ducks, geese, and so on that can be infected with the influenza virus. And of course, we have wet markets, the so-called wet markets, live markets, where many large numbers of animals are kept in close proximity to large numbers of people, allowing for the spillover to occur.
Yeah, okay, great. Next question. Any idea why the London plague dropped so quickly and rapidly? It appears that it started to drop well before the herd immunity level of 60 to 70% that we hear about.
Yeah, it's a good question. Some people have speculated. So you may have heard of the Great Fire of London that occurred, I believe it occurred in the beginning part of 1666. Some people attributed that fire to it. I don't believe that's the case. I think they've pretty much established now that the fire broke out in a part of the city that was not experiencing heavy plague. I think most likely what happened there is the plague in most pandemics appears to disappear in the winter as soon as the temperature gets cold. And I think one reason for that is that fleas don't do well at temperatures below 18 degrees Celsius. And so even in the major part of the bubonic plague back in the 14th century, most of the time the plague would begin to disappear in the winter and then maybe come back the following spring. And so I think this particular plague broke out in the latter part of the summer of 1665. And then when temperatures began to dip in the winter, it began to disappear. And then, of course, the fire happened afterwards that may have affected the ability of the plague to come back.
Yeah. Okay. How much of the mechanisms of the bacteria are relevant to viruses, such as coronavirus?
That is a very good question. The mutations that I talked about, there are a number of mutations that allowed Yersinia pestis to become a major pathogen. I don't want to pretend that I've talked about all of them. What I focused on are mutations that allowed the bacteria to use the flea as a vector because it allowed for pandemics. It allowed for the rapid spread of bacteria. Most viruses, there are some viruses that use an insect vector. I'm thinking of a yellow fever that uses mosquitoes as a vector to move from person to person. But many viruses don't rely on insect vectors to move from person to person. Those viruses, like influenza, like COVID, tend to induce coughing and sneezing as a way of moving from person to person. And so the kind of mutations that we see in SARS are not mutations that are allowing it to live inside of a vector, another organism like Yersinia pestis. These are ones that, like I said, are in the spike protein for the most part, that allow it to become more infectious. And what it appears is that these mutations allow the virus to attach onto human cells and infect human cells much better than they did before, and improve the ability of the virus to infect. So the mutations that we're seeing in the virus causing COVID-19 are different in terms of function than the ones that I talked about for Yersinia pestis.
Mm-hmm. Okay, interesting. Mass infection seems to be an ongoing natural phenomenon. As our population grows, shouldn't we be expecting more massive outbreaks in the future?
Yes, I think that's true. I think we were very fortunate throughout the latter part of the 20th century and into the current century. The last major pandemic that killed millions of people, as you might be aware, was the major influenza pandemic of 1918. Partly, pandemics are able to spread more rapidly now because the human population is much more mobile now. In 1918, the First World War was occurring, and major armies were moving around the world and allowing microbes to travel great distances very, very quickly. And then we went through a long period of time where we didn't see a pandemic that was anywhere in the same order as the influenza plague. And I think it let down the guard of some governments. As I said, scientists do scour the world and have constantly been monitoring the virus loads of different organisms, particularly in the tropics, where we tend to see a lot of viral diseases emerge. And fortunately, they have been doing that. But I think, as I said before, governments have been a little bit lax in doing what they need to do to guard for these rapid emergences of new and deadly diseases like COVID-19. If there is anything good that comes out of this pandemic, the current pandemic, is that we learn to do the kinds of things and be vigilant for these sorts of things to occur in the near future. Mm-hmm. But yeah, this is not going to go away. This is something that we always need to be on guard for.
Yeah. What are your thoughts about the ability of COVID to mutate into a more deadly form?
That's a good question, and of course, I'm sure it's keeping a lot of people up at night. Generally, viruses and other microbes that cause disease, what is, I hate to say more important to them, they're not thinking organisms, they're simply organisms that are responding to the ability to move throughout populations quickly. And what's important to them then is the ability to be infectious, the ability to get into an individual caused disease and then get out of that individual and into new individuals so that it can spread. Mutations that get selected for tend to be one that make the viruses or pathogens more infectious. Generally, this is not always true, but generally, pathogens tend to be more virulent early on in a pandemic and gradually lose virulence because it's not in their best interest to kill off victims very quickly, at least. It's more in their interest to be able to spread from person to person very quickly. So there are two properties of microbes: the virulence, which is the ability to cause damage and death, and the ability to be infectious. So generally, there's greater selection for pathogens that become more infectious, not necessarily more virulent. And that's what we're seeing with SARS-CoV-2, at least initially. It's not becoming more virulent, it's becoming more infectious with new mutations. And so on the one hand, well, that allows the disease to spread and more people may die of it because it's able to spread more rapidly. At least it's not becoming more virulent. And as far as I know, researchers are fairly confident that these new variants that we're seeing in SARS-CoV-2 are not allowing the virus to evade the vaccines that have been developing. So it appears these mutations won't affect the efficacy of the vaccines that are being distributed now.
Are you surprised by how quickly the current pandemic has spread? Has anything about this surprised you?
I guess I probably have to include myself and people being a little bit naive about this. We saw the influenza virus in 1918 spread relatively rapidly, but nowhere near the kind of speed that we saw this current pandemic. We should have been aware, and I should have been maybe more prepared for it, just at least mentally, because of the SARS outbreak that we experienced almost 20 years ago. That was caused by a virus. You may remember a SARS epidemic rose in Southeast Asia and then spread to North America very quickly. That was actually a deadlier virus than the current virus we're experiencing now, but it was not as infectious. And so we were able to contain it. And although it should have been more of a wake-up call to governments and maybe people like me, because we were able to contain it, I think maybe it gave us a false sense of security. But I too, I'm surprised that a virus could emerge and within a couple of months be able to spread around the globe and cause the kind of economic and social damage that we've seen.
Yeah, definitely. Okay, next question. Having encountered marmots aggressively going through the camp we had at Mount Whitney, can you be affected by them by contact with things they touch, or would direct contact be the only way they can transfer the virus?
This is a bacterium, not a virus. If we're talking about Yersinia pestis. And generally, yeah, I don't think you would be able to contract it through things that a rodent touch. More likely, if you were bitten by an infected rodent, or if you killed an infected rodent, got blood on you, or a flea was able to, an infected flea from an infected rodent was able to bite you. Those are the most likely ways of contracting plague nowadays. I teach a course, People's Plagues and Microbes, that talks about some of these pandemics. And one of my students a couple of years ago was a soldier who he still goes out, I think he's in the National Guard, goes out and trains in Arizona occasionally. And they have a large training area there, and they're told constantly if they come across dead prairie dogs to not go anywhere near them, because prairie dogs are very susceptible to Yersinia pestis. And as I showed you, there's a plague foci, or Yersinia pestis foci in the western United States. And so periodically we see these large die-offs of prairie dogs that you want to avoid at all costs if you happen to be hiking through Arizona or other states in the western part of the United States.
Yeah, interesting. Okay. In Peru and other South American countries, guinea pigs, which I think is called cuy, if I'm not mistaken, are eaten and often raised in a family compound for their own food use. This has been a practice for years. It is known to be a problem with disease. Is it known to be a problem with disease transfer to humans? If not, is there any scientific explanation?
I'm not aware of any. I've never heard of disease being transferred in that way. Um, yeah, and I don't have an explanation for it, except that Yersinia pestis, as we know, evolved in the old world. It did spread to the new world, and it is down into South America, but it would have only have arrived down there about 100 years ago or so. And I don't know if guinea pigs are susceptible. Some rodents, some animals are not as susceptible to Yersinia pestis as others. Prairie dogs, very susceptible. They die off very quickly. Cats, I know, are susceptible. Dogs are very resistant to Yersinia pestis. That's about the best I could do. I'm not sure why.
Yeah.
There's no disease I know of that's been transmitted that way.
Yeah. Okay. Okay. We got a slew of questions left, so I'm going to try to get to as many as I can. What do you think of the novel use of RNA for a vaccine?
It is a tremendous leap forward. One of the most astounding things that has come out of the pandemic for me is not only how rapidly it spread, but how quickly we were able to come up with an effective vaccine. I was watching, I think it was 60 Minutes a few weeks ago, where they were talking about the fact that it literally took 2 days to design a vaccine. Once we knew the structure of the spike protein that's shown in this picture, I don't know if you can still see the picture here.
Yep.
The vaccine targets this spike protein. Once we knew the structure of that spike protein, we were able to, or not me, but the researchers were able to design a vaccine, an RNA molecule, within 2 days. And to synthesize that RNA molecule, we can synthesize RNA molecules in machines now. And then, of course, it's a matter, the hard part is to scale that up, put it into some sort of carrier that allows it to be injected into somebody's arm, and make a vaccine. So this is a game changer to be able to design a vaccine so rapidly and move into the clinical testing stages. To be able to compress that all down into less than 12 months has been absolutely remarkable. Most vaccines that I know of, the traditional ones, generally take 3 to 5 years to go from conception to demonstrating that they're clinically safe.
Yeah, it's been fascinating to watch all of that. Why don't dog and cat fleas propagate plague?
I think I'm not. I think they can. The dog, there's something about the dog. I don't think it's completely resistant to bubonic plague, but even if it's been shown that something like 2,000 different species of fleas can carry bubonic plague. But some of the animals that are bitten by the fleas have higher resistance to the plague. Dogs apparently have very high resistance. Cats, not so much. Yeah. Just as an aside, the Great Plague that struck London in 1665, some people think it was made worse by the fact that the city decreed that all cats and dogs should be killed. So many cats and dogs were put to death because it was thought for some reason that they were helping to spread the plague, when they were probably actually holding back the plague by eating and destroying the rats that were carrying the fleas.
Yeah, interesting. Okay. Why was polio not considered a pandemic since it has been in multiple countries and continents?
I don't know. I'm not sure that it has never been considered as a pandemic. It was worldwide. It's not so much now. It's confined to more underdeveloped countries. I'm not sure that it doesn't qualify as a plague of some kind. I'm not sure of the numbers. That would be something. I mentioned I teach a course on people's plagues and microbes, where I've looked at a number of different diseases and pandemics. I've never looked into polio, so I don't have a good idea of the number of people who've been infected or died, but I don't see why it wouldn't qualify as a pandemic.
Yeah. Okay. How dangerous are pets, particularly dogs, specifically emotional support dogs, to transfer COVID?
Uh, I haven't heard much about that lately. That I know early on in the pandemic, there was at least one or two cases that indicated that uh, the virus could be contracted from a dog, but I haven't heard that lately. And so I think there may have been just a few isolated cases. I can tell you, if that's possible, there's a lot of people in my neighborhood that are ignoring it. There is an explosion of puppies in my neighborhood where people that are sequestered at home in the pandemic are finding support in canine pets. And I have to include myself in that. I have a 4-month puppy around here somewhere that I've had a lot of enjoyment of, even though I'm restricted to my neighborhood now.
Very good. And what type of puppy is it?
A golden retriever, a
Oh,
very
perfect.
naughty golden retriever.
Perfect. We welcome that. We have a family dog called Darwin, who's a golden retriever.
Perfect name. Perfect.
Yes, perfect. Yes. Okay. Let's see. Last question. The COVID came from the animal pangolin as well as bats? Question mark.
So that's still up in the air. I think there's about 39 base pairs in the RNA genome of the SARS-CoV-2 virus. The vast majority of that sequence is very, very similar to the viruses that live in bats. Interestingly, though, there is a small region of the gene for the spike protein. So the spike protein has its own gene in the virus, the SARS-CoV-2 virus. And when that short region of the spike protein gene is sequenced, it's most similar to a SARS virus from pangolins. And as far as I know, nobody has a good explanation as to why that is. It may just be coincidence, or some viruses, I don't know if it's possible for SARS, some viruses, if you have two different viruses but related viruses infect the same animal, sometimes they can recombine into new forms of a virus that can spill over into other species. That certainly happens with influenza, where avian and mammal forms of the virus, if they infect the same animal, spit out viruses that are combinations of the two that are brand new. That's what happened in the 1918 flu pandemic. A brand new virus that was a combination of a mammal and avian virus emerged and was able to infect human populations. So I don't know if that's what's happened. That's one of the things that needs to be answered going forward.
Yeah, great. Well, Dr. Bird, thank you so much for this really fascinating and obviously very timely topic. We really appreciate your time, and thank you for kicking off U3A and giving us a great boost here to the program. So we appreciate all of your time and talent and expertise today.
Well,
so go ahead. Yeah.
Thanks, Nicole. I have to tell you, this was a lot of fun. I really enjoyed this.
Yeah. Yes. Well, perfect. Well, we hope to have you back in a different session with hopefully talking about COVID being in the past for us, but maybe talking about some of the other topics that you're an expert in as well. So.
Okay.
Great. Well, I have a few announcements to make to the group. I'll allow you to get on with your day and enjoy that puppy.
All right.
All right. Thank you again so much for your time.
Take care.
Thank you. All right, everybody. Great first day. Thank you so much for that. I see some applause and some thumbs up. So hopefully you enjoyed that presentation. Just another couple of reminders, just so that we are all on the same page. Later today, you'll receive an email from me that has the recording from today's session, both the technology workshop and then a separate file for Dr. Bird's presentation. A couple of questions I've had was, can I share that presentation? Yes, you can share that presentation with your friends. I would just ask that you don't share all the presentations, because we would like that person, your friend, to be participating in U3A as a paying participant. So feel free to, share one or two of them, but let's try not to keep them out into the world where others get it for free and now you've had to pay for it. So look for that. And then reminder about watching Fandango at the Wall. If you haven't already done that today, please make sure you do that, just because, as I said before, I think you're going to find the discussion tomorrow to be much richer if you have a sense of what the documentary is about. And with that, I will bid you adieu for today. Reminder that technology starts at 8:15 in the morning. I'm happy to stay on for a few minutes if anybody has any specific questions for me. Any questions or comments for the group?